Hello, everyone. We'll be starting in a minute. This is going to be what we call a Simple Science Snippet, and it's part of a series that we'll be introducing everyone to. And each time we present something, we'll be having a different topic and a different expert who will be leading us. And so it's now 7, so let me introduce myself. So my name is Judith Zelikov, and I am a toxicologist in the Department of Environmental Medicine at NYU Grossman School of Medicine. And I'm also the Director of Community Engagement for the NYU National Institute of Environmental Health Sciences course center. And so I lead the community engagement core with my co-lead, Simona Kwan, also from NYU Medical School. And so what we'd like to, and so again, this is in collaboration and in support with many of the communities that we work with in Brooklyn, in New Jersey, and so we're putting this on live for you. So I'd like to introduce, of course, my, okay, I'd like to introduce Dr. Harati, and he is going to be the lead for today. We're hoping that this will be more of a question and answer live broadcast, so he'll be able to answer any questions that you may type into the chat, and I'll be reading them to him. If there are no questions, then he will continue to talk to you about COVID and vaccines. So Dr. Harati is a first-generation immigrant who grew up in the Midwest. He did his medical training at Washington University in St. Louis and went on to be an infectious disease specialist at the University of Pennsylvania. After his training, he also spent 7 years studying the immune system and how vaccines and the immune system interact. He's currently an assistant professor in the Division of Infectious Diseases and Immunology at NYU Grossman School of Medicine. And he's a faculty member in the Vaccine Center at NYU, where he's part of the COVID vaccines clinical trials. Right now, his research laboratory works to understand how our immune system and our immune responses to vaccines change, and how things like aging, inflammation, other diseases influence these vaccine responses. So without any further ado, I'll turn the program over to Dr. Harati. And please remember to type in your questions. We're hoping that for the next 15 minutes, you'll be informed and get your questions answered on this very imminent disease. Thank you, Dr. Harati.
Hi, good evening. Thank you so much for the invitation and for letting me be part of this great event. It's really terrific to be able to have this conversation and to be able to answer questions that people might have about the COVID vaccines, COVID disease and or the vaccines, depending on whatever people are interested in hearing about. So I'm just going to start talking about the COVID-19 and the process of how vaccines are made. But again, please feel free to interrupt if you have questions, and we'll take it from there. So of course, we've been dealing with this COVID-19 pandemic now for so many months. We've been dealing with this since it was first discovered back in November, December last year. When we first heard about it was January, February. And of course, since then, it's had a terrible toll on society and our way of life in general. Fortunately, it looks like there's at least a glimmer of hope on the horizon, that we have these new vaccines potentially on the horizon, and they look like they're quite promising. But of course, not everybody knows a lot about these vaccines because they're so new. So we thought this might be a good opportunity to talk about some of these COVID vaccines and, what are they, how do they work, and, what risks are there? Could they make you sick, and so on? again, with the caveat that these are still fairly new, again, I thought it might be helpful if I share with you some of what we've learned along the way about these COVID vaccine candidates. So I'm here with Layla, who's going to help me out. But here we have a model of a coronavirus. Obviously, real viruses are much smaller than this, but the coronavirus is small. It's, roughly it's round, give or take. And on the surface of the coronavirus are these things called spike proteins, right? So this is something you'll hear about over time if you haven't heard about it yet. So that's part of what gives coronaviruses their name. Having these little spikes almost makes it look like there's a crown on the virus, which is how it gets its name, coronavirus. So these spike proteins are actually what the virus uses to get into our cells and to make us sick. So as people started to think about how do we come up with a vaccine to protect us against this illness, COVID-19, the first thought was, well, why don't we get rid of or neutralize the part of the virus that is used to get into our cells? So in other words, the target of all of these vaccine candidates right now is the spike protein. So with different ways of doing this, such as, for example, if this is an antibody that your body is making, we can neutralize some of these spike proteins, right? So we can decorate or basically coat all these spike proteins with little clips, and that neutralizes them, and that keeps the virus from getting into our cells and making us sick. So, okay, we have this tool. This is great. Our bodies normally make these proteins called antibodies to help protect us against things like other viruses and bacteria. But now it's not just the case that we want to have these, but we want to have them before we ever see the virus, right? We don't just want the body to react to the virus and make these proteins, but even before we get the virus, we want to have these antibodies in place so that the virus never has a chance to get in to make us sick. So the way these vaccines work, so right now in the news, you're going to hear about these vaccines from the companies like Moderna and Pfizer. They're the ones that are most often in the news right now. Both of these companies have a vaccine candidate that's pretty far along in development. These are called mRNA vaccines. And essentially what that means is if you take the virus, so they don't use the virus, it's not a live virus vaccine where they, try to take this virus and weaken it, for example, but rather they take just the message encoding the spike protein. Let's say here is the message. This is a rolled up piece of paper, but let's say this had the code for the spike protein, and they put that into a lipid nanoparticle envelope. And this is what is actually injected into a recipient. And with 2 injections of this 1 month apart, the goal is to then teach the body to now make these antibodies without ever having to run into the virus itself or to have any chance of making you sick. So the bottom line is that both of these vaccine candidates, both Moderna and Pfizer's vaccines, are based on this idea of messenger RNA or mRNA, where we just bring the spike protein instructions into the body and we teach the body how to make antibodies to neutralize the spike protein.
So Dr. Haradi, we have some questions, if
Please.
now is a good time.
Please go ahead.
Okay, so question, when do we expect the vaccine to be available for the general public?
Yeah, that's a great question. I would say this is something all of us want to have happen as soon as possible, within the bounds of safety. I think that's really the crux of it, is we want to make sure that what we're doing is safe. At this point, a lot of these vaccines have been trialed in volunteers. So for example, at NYU in the Vaccine Center, we've been part of both the Pfizer vaccine studies and now we're part of the AstraZeneca vaccine studies. And so for Pfizer, Moderna, these are the vaccines you're hearing about. For each of these, over 30,000 people for each of these trials have enrolled in these vaccine studies. So in terms of when are these vaccines going to get rolled out, right now, Pfizer and Moderna have both applied to the Food and Drug Administration, to the FDA, to get approval for their vaccines. Obviously, the FDA hasn't given approval yet, but it's expected emergency use authorization will come before the end of the month. Right now, different groups are meeting to discuss what the right rollout strategy would be. Or just earlier this week, the ACIP, which is the committee a 15-person committee that advises the CDC on what its guidelines should be, they met and they had a public meeting to discuss what the phased rollout should look like. Again, I think there's a lot of uncertainty here. We're not sure how fast these companies will be able to make these vaccine doses and how we're going to get these distributed. And, is this going to be available at your local pharmacy, or is it going to need to be through your physician's, your doctor's office? Are they going to have the right freezers and so on? But I would say that probably the first people will start to get the vaccine before the end of 2020. And I think the broader general rollout's likely to happen probably in the first quarter of 2021. But again, that could very well be delayed if there are problems with manufacturing or delivery of these vaccines.
Thank you. So we have another question. If I had COVID-19 antibodies because I had COVID, do I still need to take a vaccine?
Yeah, that's a great question. I would say possibly, probably, although the truth is that's not a question we for sure know the answer to yet. If you've had coronavirus infections and your body has already learned to make these antibodies, well, you have antibodies then in your system. What we don't know is quite how long those antibodies are going to last in your system. Some cases, antibodies are in your body for years and years and years, whereas for other instances, your antibodies may not last nearly as long, and you may need a vaccine to boost up those antibody levels. So I think that remains to be seen. Right now, I'm not sure we have the answer to that question of whether we need to give the vaccine to people who've had coronavirus COVID-19 infection already. But again, hopefully we'll have the answer to that soon.
So from a conservative perspective, would you say if you were the one with testing positive for antibodies, would a vaccine cause detrimental effects if you decided to get the vaccine?
So I'll give you two versions of an answer. My opinion is I don't think, based on what we know now, I don't think there would be a detrimental effect if you'd had COVID and got the vaccine. I think the pharmaceutical companies will have the answer to that, because a lot of these studies, in these 30,000 person studies, one of the aspects is that they get blood on the participants at the very moment that they start enrolling in the trial. So there may be people who had coronavirus infection, didn't realize it, or maybe had asymptomatic infection, who are enrolling in these trials, getting the vaccine. We haven't seen any safety signals, any adverse events that are sort of out of the ordinary, out of proportion. But I would say that the drug companies will be able to give us some information about this in due time once they sort of compile all the data. But again, my knee-jerk reaction, my guess is that there won't be any harm if you've had COVID to get a COVID vaccine.
Okay, we have a few more questions popping in. So we're hearing from community members that this vaccine is rushed and it's not safe. So how can we go about alleviating some of these fears?
Yeah, I mean, that touches on a really important point. Again, we want to make sure, the medical community, we want to make sure that these products that we're giving, whether vaccines or therapeutics, drugs like remdesivir and convalescent plasma, we absolutely want to make sure that we're not causing any harm when we give these, that we're doing something that is to the benefit to the individual, not necessarily, going to cause them problems. As far as whether this is rushed or not, I think that's a bit of a tough question. And the reason I say that is because, typically vaccines have taken years to develop. So when you look at it from that perspective, if you consider that, let's say for the original SARS-CoV virus, SARS, in other words, that we had back in the early 2000s, we don't actually have a vaccine. And now we're probably, 17, 18 years out from that virus infection, when that virus first emerged. For other things like polio, it took like 50, 60 years until a vaccine was developed. But now we're also talking about that we're in the 21st century and a lot of our methods are a lot faster than they used to be. Moderna, I believe, had designed its vaccine within just a couple of days. They knew what they were trying to do, which was to take the spike protein and put it in a lipid nanoparticle and deliver this as a vaccine. So I would say that, for those initial steps often take years, those initial steps of figuring out the strategy, how we're going to make a vaccine in the first place. Those have in the past taken years or even decades. That part went a lot faster. But I would say the key part of this is the safety piece. This is why we're doing these 30,000 person trials, where we are blinded, which means neither the study trial, the physicians doing the studies, nor the participants know if they're getting the vaccine or the placebo, because we want to have the best perspective. We want to have sort of an unbiased look at whether this is going to cause side effects, adverse events in some people over others. So I think that's the part where the FDA is going to take its time, and they will review all the safety data, and I don't expect them to approve a vaccine that has serious risks.
Okay, so I think we're going to just take one or two more questions, and then we'll wrap this up. There's a question about, can you talk a little bit about reinfection? We're reading in the news that people are getting this more than once. Any suggestions, any insights?
Yeah, I think this is a big concern. Obviously, getting the infection the first time is bad, and getting it second time is just a terrible thing. Clearly, there have been some very well-documented instances where people have gotten infected more than once. We can look at the actual virus that they have, what they're infected with, and we, in other words, the medical community has demonstrated that they were two similar but slightly different coronaviruses that they got, just different enough. They're still, all of it's still COVID-19, but they're different enough that we can say it wasn't the virus they had originally. So I think that is likely is happening. Clearly, again, there have been some cases. I don't know how often it happens, but I think it's just a reminder that we're not out of the woods yet. And even for some of the people who've unfortunately had COVID-19, it's still really important to take all the precautions to limit your social interactions, to wear face masks, especially when you can't socially distance, but really anytime you're indoors or around other people, until we really have good vaccines and have tamped down this infection, I think we're all still in a bit of danger.
Another question, are the messenger RNA vaccines that you spoke of, are they safer than the traditional vaccines of a weakened or dead virus or attenuated virus?
So I think the word safer is the part. Let me start with that. So obviously, again, when we're giving a vaccine, we're delivering a pharmaceutical drug to a person who's otherwise healthy, who's not sick with the virus or sick with anything, an infection of that sort. So there are sort of different kinds of reactions that people can get after a vaccine. There are reactions that, in a way, you could say we almost expect them. So for example, if I get the flu shot every year, when I get the flu shot, my arm is often sore, and it's sore for a day or two. So that's a side effect. That's an adverse event due to the vaccine. Sometimes you might get some swelling or some redness. This all goes away. So in other words, there's a class of side effects that we call reactogenicity. Every once in a while, people will also get fevers or chills. We've seen all of these reactions in the COVID vaccine studies that we've been doing at NYU, where again, even though we're blinded and we don't exactly know who's getting which product, the fact is that people are reporting fevers and chills sometimes. And we think that that's perhaps due to them getting the vaccines. But all these side effects tend to last about a day, maybe two, and they go away pretty quickly, and they're relatively mild. So are these vaccines, mRNA vaccines, safer, was the question. So that's, I think, to be seen. I think we don't have enough safety data yet, at least not data that's published that I'm aware of, to be able to really compare vaccines to one another. But I would say, our vaccines generally have a very good safety profile, at least the vaccines we're using today. Our flu vaccine, tetanus vaccines, hepatitis vaccines, we use these regularly. Tens of millions, if not hundreds of millions of people have gotten these vaccines. And in some cases, like the flu vaccine, people are getting these annually. And I would say these vaccines are extremely safe. And likewise, that's the bar for these mRNA vaccines. Yes, we're in a pandemic, absolutely, no question about that. But at the same time, these vaccines need to be safe. And, we have to make sure we've got enough data to guarantee that people are going to do well with these vaccines, or else maybe we should not be using this vaccine and we should go to another technology, another platform altogether.
Sorry. We have one final question. I know the sample size of 30,000 is larger, but isn't the number of infections too small for a strong recommendation of efficacy?
So the studies are 30,000, although Pfizer also expanded its study to 44,000. So they're enrolling, a lot of people. I think at the interim analysis, what was published or what was released about a few weeks ago, was that they had 100 and something people who got infected out of the 30,000. 30 to 40,000 in their trials. Both Moderna and Pfizer had these numbers. So essentially what's done here, sort of before the trial ever starts, is that criteria are set down, statistical criteria for what level of confidence are we going to need before we can make a claim that the vaccine either works or doesn't work. And so that's essentially coming down to the statistics of, how many cases in the placebo arm versus how many cases in the vaccine arm would it take before we had some confidence that we're not just seeing something happening by chance, but in fact, there's a consistent effect. Both in Pfizer and Moderna's cases, essentially for about 100, give or take, I can't remember the exact number, but for about 100 infections in total that they saw in the vaccine study, only about 5 to 7 of them were in the people who had gotten the vaccine, and 80 to 90 of those infections were in people who had gotten the placebo. So it's statistically unlikely that that's due to chance, and that met the criteria that they had established ahead of time by which they could use to announce these results.
So I will take the liberty of being the moderator to ask one last question. And also, I think there were so many questions and so much interest in this that we may be asking you back, Dr. Harady, for a part 2 of this. But my question from me is there are Moderna and Pfizer are only 2. I mean, there's AstraZeneca, there's other companies. What is the difference between their vaccines, and how do we know what we're getting and what we should choose?
Yeah. Right. So, we obviously would love to have head-to-head studies to compare these vaccines, because we want to, everyone wants to get, we should all want to get the best vaccine that we can, right? So that's again, a piece of information that we don't have yet, is how do these vaccines compare? The different companies are approaching this problem in different ways. So whereas Moderna and Pfizer are using this messenger RNA-based platform, AstraZeneca, for example, is using a different idea. They are also using the genetic code for the spike protein, but instead of delivering that in a lipid nanoparticle, they're actually using a different virus, the shell of a different virus, not any of the other parts that make the virus a virus, but just the protein shell. And that's how they're delivering the spike protein genetic code. And again, there are a number of other companies, and they're doing it also slightly different ways. So, the end result, the end goal is to generate these antibodies, these protective protein responses that will protect us against the virus. Again, I think time will tell if one is necessarily better than the others. So far, at least in the phase 1 studies, the preclinical data, all of these vaccine candidates that have made it to phase 3 have been able to induce antibody responses pretty robustly. All of them look actually pretty good. And that's really great, because we're not going to be able to do this. We're not going to get everybody in society vaccinated with just one platform or just 2 companies that make it. We really are going to need, a lot of winners in this race, if we have a hope of vaccinating as many people as we can in as short a time frame as we can.
So we have one last person who, only if Dr. Harady is willing to stay a few extra minutes to answer this.
No worries. Yes, go ahead.
Thank you. So are there any possibilities of the mRNA vaccines affecting our DNA in the long term, say 5 years down the road?
Again, with the caveat that we don't have 5 years of data on these vaccines, I would say that's really unlikely. These mRNA vaccines, they don't stay in the system. They don't stay in the cell. The mRNA is there for a brief period of time, and then your cell chews it up and degrades it. The mRNA doesn't turn into DNA, and it doesn't integrate into your DNA, and it doesn't last in a way that persists. And so for that reason, I would say that its effect really is going to be to just help make these anti-spike antibodies. And those antibodies are the thing, again, that the goal that we're shooting for. But again, the caveat is, I think we are going to need more data before we can really confidently say, if there are any longer term risks. But again, so far, we haven't seen differences in adverse events between the vaccine and the placebo arms, at least that we know about.
Well, thank you. I have to say that we've enjoyed watching Lael with the antibodies on her fingers. And I hope that everyone who's listening in has asked their questions. I know there's probably lots more. We'll ask Dr. Haradi to come back. Maybe there'll be more information on the vaccines in January that we can talk about. And I thank you all again for listening in, and please let your friends and family know that it will be online for quite a while, and you can catch it, maybe not live right now, but certainly can see it at any point. So thank you. Thank you again. Thank you, Dr. Haradi, for an excellent job. And everyone, be safe. Thank you.
It went